you consider what it takes to produce complicated structures like living organisms or buildings, then it's obvious that you need a plan. If we're thinking about buildings, then we've all got some idea about what this means. Consider a building like this one. The architect has an idea, he converts that idea to a series of plans, and the builders acting on those plans produce the final structure. Now, when we're considering living organisms, what do we mean by a plan? Is there anything at all analogous in the egg? Any sort of design that tells the organism how to develop? Now, this isn't a new idea, nor is it a new problem. People have been puzzling about this literally for thousands of years. And some of the early ideas nowadays seem, well, a little bit naive. For example, it used to be thought that inside the egg there was a tiny adult. So in a human egg, you'd have a tiny man, which is sometimes called a homunculus. So all the development involved was growth. The little man grew, and out he came as an adult. Well, those ideas may seem fairly silly to us these days. But that's all very well, but what sort of answers do we have? We're still left with a question. So once again, is there anything about the egg, any design, any chemical perhaps, any structure, which tells us, or at least tells the egg, about how the embryo is supposed to develop? A clue comes from the study of two marine organisms, Ilianassa and another mollusk displaying similar features during early development, Dentalium. The fertilized egg of Dentalium. Is this a 3-cell embryo? Well, whatever it is, we can watch its formation. To do so, let's look at Ilianassa. It's much the same. As the egg enters the first cleavage division, a portion of cytoplasm buds out. This bud, the polar lobe, is not a cell. It has no nucleus, just cytoplasm. So the shamrock shape, called a trefoil, is two almost completed cells, plus a cytoplasmic bud, the polar lobe, attached to just one of the forming cells by a narrow cytoplasmic bridge. As the first cleavage nears completion, the polar lobe is resorbed. Look at the embryo at the top. Resorption is into just one of the two cells, the one to which the lobe is attached. Let's take another look at resorption. These time-lapse techniques enable us to see clearly what is happening. Well, the result is a 2-cell embryo, with one cell considerably larger than the other, larger because it resorbed the polar lobe cytoplasm. Remember, the polar lobe contained no nucleus. As the embryo continues to develop, further cleavage divisions occur. At the second cleavage, a polar lobe is again extruded. Subsequently, this too is resorbed into just one of the cells. So ultimately, this gives a 4-cell embryo. So in going from a 1-cell egg to a 2-cell embryo, the organism passes through a trefoil stage. But why? Why go through a trefoil? What is the function of the polar lobe? One way to find out is to chop it off. The larvae developing a few days later look like these. Quite different from the normal larva on the top left. Those from lobeless embryos have several tufts of cilia. Also, they lack several important structures. No eyes, no beating heart, no shell. These abnormal larvae swim around for a few days and then die. So presumably something in the cytoplasm of the polar lobe must normally seem to instruct the cell into which it's resorbed how to develop. Early development in a quite different organism, an insect, the midge, Smitia. Nuclear division is occurring, but as yet there are no separate cells. The nuclei migrate to the periphery of the egg, and then they're packaged off by cell walls to give a layer of cells, the blastoderm layer. Whereas at the posterior pole, on the right, a separate group of cells, the pole cells, are being formed. Formation of the blastoderm layer is followed by the rapid movements of gastrulation. Let's now slow things down and take a much more detailed look at the very early stages, this time in the fruit fly Drosophila. Just the posterior pole of an early embryo. It's at the nuclear division stage and there are very few cells, but some pole cells at this end. These gradually form, but there's still no blastoderm layer. Now the blastoderm's apparent, that single layer of cells around the periphery, and off to the right at the posterior pole, a bunch of pole cells. Later still, when the blastoderm is fully formed, we can see this distinct nest of cells, the pole cells. In this rather beautiful scanning electron micrograph of a whole egg, the pole cells are very obvious, at the posterior end again. Well, that's how pole cells form, but what are they? If we take a very early embryo, before the pole cells are formed, and irradiate just the posterior pole of the egg with ultraviolet light, pole cells fail to form. However, the embryo goes on to become an apparently normal adult. Normal looking, but sterile. So, irradiation of the posterior pole causes both loss of pole cells and sterility. So pole cells are the progenitors of sperm and ova. Presumably, irradiation destroys something in the posterior pole cytoplasm that normally induces pole cell formation. This is confirmed by an experiment by Carl Ilmenzey. Ilmenzey started his experiment with a newly fertilized egg of Drosophila. He then withdrew cytoplasm from the posterior pole of that egg and transferred it to the anterior pole of a second egg. He used eggs from genetically distinguishable flies. We've color-coded those eggs. The second grey egg was then allowed to develop, and it was found that at the anterior pole something abnormal was occurring. Cells were developing which looked remarkably like the pole cells which were developing at the usual posterior pole. This looked as though the cytoplasm from the first red egg had induced the formation of pole cells at the anterior pole of the second grey egg. Appearance isn't enough. So what Ilmenzey did to further test that these cells were in fact pole cells, in other words the forerunners of germ cells, was to take these cells from the anterior pole of the second grey egg and transfer them to the posterior pole of a third egg. The third, yellow egg, was then allowed to develop. And finally, it gave rise to an apparently perfectly normal-looking adult. The fly from the yellow egg, which we've also colour-coded yellow, was then mated with an ordinary natural yellow-type fly, and the offspring were examined. Some of them were like the parents, that is yellow, but others were grey. This means, presumably, that in the adult yellow fly, the one which came from our experimental yellow egg, some of the sperm or ova, depending whether it was male or female, were of the grey type. Now, the only place they could have come from was from those cells which we transferred from the anterior pole of the second grey egg. And of course, those cells were induced out of position from the transfer of cytoplasm from our original egg, that is, the red egg with which we started. So it looks as though something in the cytoplasm at the posterior pole of the original egg is capable of inducing pole cells in abnormal positions. That is, there are factors in the original cytoplasm which can control the formation of pole cells. Do factors of developmental importance only exist in mollusks and, in the case of insects, for pole cells? Well, let's look a little later in the development of an insect, picking it up where we left off, during gastrulation, once again in Smithia. Here some of the structures are a bit more familiar. The larva is beginning to form. The organism starts to take on a sort of segmented shape. It goes through a stage known as the germ band. Now there's an appearance of a head and a tail end. It's beginning to look much more like an insect larva as we normally know it. How, if developmental information exists in eggs, can it program something as complex as this? Not just a few pole cells, but something as complex as a segmented organism with a head, tail, and of course internal organs. That's the key question. So what sort of information could it be? Think of an egg and imagine that at the anterior pole we have the source of a chemical, and the chemical travels from this source down the axis of the egg. So we have a gradient of chemical down the egg. In principle, this gradient could be used to specify particular structures to be made at particular points in the egg. From a variety of experiments on insect eggs, it seems that in fact we need to postulate two such gradients. At the other end of the egg, another source, and once again a gradient of substance, this time travelling up the egg. So as a whole we have two sources, both giving out substances but different ones. In this way we have a double gradient in the egg, and this could act as a source of information. At any particular point along the axis, the ratio of the two substances could specify which structures would be formed by the cells which gradually appear in the egg as it develops. Now think what would happen if we were to knock out one of the two sources. If the gradient model is correct, we might expect this to drastically affect the embryo as a whole. Smithia again. But in this case, the very early embryo was irradiated with ultraviolet light at the anterior pole. At this stage, things look like the normal developmental sequence that we've already seen. But watch what it looks like as development proceeds. In particular, look for a head and a tail. It's pretty obvious that you're not going to find a head. This organism, which is known as double abdomen, has got two tail ends, apparently arranged as a mirror image duplication. So it seems that the ultraviolet irradiation of the anterior pole, just the anterior pole, has upset the whole organisation in the egg, consistent with our hypothesis. So here again, it seems that developmental information does indeed exist in eggs. But how does it get there? Back to a marine organism. This time, a seaweed. The egg of Fucus hours after fertilization. At 3 days, there's an obvious protuberance, the rhizoid, by which the growing embryo can attach to the substratum. This rhizoid also indicates a distinct polarity in the embryo. By 7 days, it's somewhat larger. The orientation in which the rhizoid grows out from the fertilized egg can vary. For several hours after fertilization, this orientation can be affected by such external factors as heat, light, or pH. For example, if a newly fertilized egg is illuminated, the rhizoid grows from the shaded side. What sets up this polarity is not yet known, but it seems that at the time the axis is fixed, the eggs can generate electric currents. This implicates ion movements in the fixing of polarity. Once fixed, the axis of polarity determines the direction of rhizoid growth irreversibly. Ionic movements, particularly of calcium, have been implicated as important early events in other eggs. The eggs in question are from Medaka, a tiny freshwater fish popular with children in the Japanese countryside. It's even sung about. But the experiments were done thousands of miles away. Professor George Reynolds, Princeton University.
What we're doing here is looking for the very first act of the fertilization of the Medaka egg. The Medaka is a Japanese fish, very popular in laboratory experiments, and it has been noted that when fertilization starts in this egg, there is a vesicle change in the egg outer membrane. It's also been hypothesized that there's a calcium release. Now, it is possible to inject the egg with a photoprotein such that that calcium release would give out light. The characteristic of the light, though, is that it is so weak that it can't be seen by normal observation or conventional photography. So that this apparatus connects a microscope to an image intensifier, which has the property of giving out 1 million times as many photons as it receives at the input end. These photons hit a phosphor, which is very similar to a television screen or a cathode ray oscilloscope screen. That is sufficient light for a Vidicon to pick it up and then transmit to a tape recorder and television monitor.
So there it was, the egg under the microscope, a barely visible grey circle, not very impressive. And would the experiment work? Would anything happen when the sperm was added? And would they be able to see it?
Wow, that is, it started at a tiny little spot. Look at it go, man. And you can see a ring? You really can, you really, really can. Fantastic. Oh, that is beautiful. Fabulous. Oh, I wouldn't touch it. I won't touch it yet. Oh, that's gorgeous. It's a halo. Yes. Isn't that beautiful? Green. Look at it spread. Oh, delicious. I won't touch it yet. In a while, it may require some focus. Oh, that is not.
Let us take another look. Where the sperm contacts the egg, calcium is released, made visible to us by the excitation of the photoprotein. The calcium, and hence the light, gradually spreads over the surface of the egg. What this means, no one can be sure. But at least it indicates a wave of polarization as an early event after fertilization, an event that possibly helps polarize the egg for future development. So in fish, mollusks, seaweed, insects, there is some evidence for factors in very early embryos, in some cases present even before fertilization, that influence much later developmental events. These factors may help determine the development of specific cells or organs, Or they may be of a more general graded nature, for instance involved in the overall head-tail polarity of the organism. Exactly what these factors are and how they operate is yet to be discovered. What is likely is that in order to answer these crucial questions, developmental biologists are going to have to delve further and further into the earlier stages of development.
But you can still
It's
see
still
it
pretty
there.
well in focus.
Yeah, I'll leave it.
The egg itself may be helping focus it. Because it looks like it's getting an awful lot right. Well, maybe.
It's very, very clear.
Of course, this may be coming back up.
What?
You're right. You don't know. You don't know.
I
You've
think
never
we do.
seen that.
So you reflect off the end.
What
Travels
happened?
in two directions.
Now it's at the end.
Well, you know it doesn't do that.
Of course we know it does.
From the side.
It started up here. That's just about where the antipodes should be.
Yeah, that's perfect. And now it's nothing else. That's right, applause at the end of the show.